Meiosis is a type of cell division that results in 4 daughter cells, each with half the number of chromosomes of the parent cell, as in the production of gametes and plant spores. Prophase. Within the nucleus of the cell, the DNA coils into chromosomes and becomes visible. These chromosomes are in diploid cells, meaning that one set of chromosomes is inherited from the mother and one from the father. The homologous chromatids pair up into chromosomes from the maternal and paternal chromosomes, and they cross over. This allows chromosomes to exchange genetic material. This is called synapsis, the fusion of chromosome pairs. Chiasma is the point of crossing over between non-sister chromatids. Recombination is when the two molecules of DNA exchange pieces of genetic material. The pair of chromatids are connected through the centromere. The nuclear membrane begins to break down as the spindle apparatus forms. Metaphase. During metaphase, the chromosomes line up in pairs along the equator. Each pair attaches to a spindle microtubule at the centromere. This increases genetic diversity. Independent assortment dictates the position of chromosomes, and it is the random assortment of DNA that forms the gametes. Anaphase. The spindle microtubules contract towards opposite poles of the cell, separating the homologous pairs and half the chromosome number, 23. The cell is changed from a diploid to a haploid. The cell begins to elongate, then pulling apart of these chromosomes is called disjunction. Telophase. The nuclear membrane begins to form around the new nuclei. The homologous chromosome pairs complete their migration to the two poles as a result of the action of the spindle. Now a haploid set of chromosomes is at each pole, with each chromosome still having 2 chromatids. A nuclear envelope reforms around each chromosome set, the spindle disappears, and cytokinesis follows. In animal cells, cytokinesis involves the formation of a cleavage furrow, resulting in the pinching of the cell in 2 cells. After cytokinesis, each of the 2 progeny cells has a nucleus with a haploid set of replicated chromosomes. These cells are genetically unique to the mother cell, but have half the number of chromosomes of the parent cell. The purpose of this first phase of mitosis is reduction division, the first cell division in mitosis, the process by which germ cells are formed. In reduction division, chromosome number is reduced from diploid, 46 chromosomes, to haploid, 23 chromosomes, also known as the first meiotic division and first meiosis. Prophase 2. Within the new 2 daughter cells, new spindle microtubules begin to form. The chromosomes recoil. The nuclear envelope begins to break down. Metaphase 2. Individual chromosomes line up along the equator of the cell. The nuclear membrane fully breaks down. Spindle fibers attach themselves to the chromosomes at the centromere and pull them away. Anaphase 2. The centromeres separate and the 2 chromatids of each chromosome move to opposite poles of the spindle. The separated chromatids are now chromosomes in their own right. Telophase 2. A nuclear envelope forms around each set of chromosomes. Cytokinesis takes place, producing 4 daughter cells, gametes in animals, each with a haploid set of chromosomes. Because of crossing over, some chromosomes are seen to have recombined segments of the original parental chromosomes. A polar body is a small haploid cell which does not have the ability to be fertilized. When certain diploid cells in animals undergo cytokinesis after meiosis to produce egg cells, they sometimes divide unevenly. These 4 genetically unique daughter cells in a female are called ovum. They can be fertilized with a sperm, allowing a female to reproduce. During meiosis, many errors can be made. Non-disjunction is when homologous chromosomes or sister chromatids do not separate properly during meiosis. This can occur during anaphase 1 or anaphase 2, when the chromosomes are being separated by the spindle.